Welcome to the CIBM Center for Biomedical Imaging Breakfast in Science Seminar number 5. It is my great pleasure today to introduce you to our speakers. Ileana Jelescu, who is a research staff scientist and 14T MRI operational manager at the CIBM MRI EPFL Animal Imaging and Technology section. Ileana will be sharing with us how the MRI scanner can be seen as a sharp in vivo microscope from white to gray matter. Thank you, Ileana. The floor is yours.
Thank you, Pina, for the introduction, and good morning, everyone. So it's a great opportunity, indeed, for me to present my work, and I wanted to explain here how we can use the MRI scanner as a sharp in vivo microscope in order to infer things about the microstructure in white and gray matter. So first of all, can we hope for a tool that can achieve in vivo non-invasive microscopy? So all of the current tools actually cannot rise up to this pretense, and of course even two-photon microscopy remains fairly invasive and can only achieve limited depth. However, if we did have something that could have in vivo non-invasive microscopy, it would be applicable to both human and animal studies, and would allow longitudinal study of damage and repair, as well as enable fine diagnosis of disease or injury, and monitor evolution and response to therapy. And so the answer I hope I can give to you today is that indeed, yes, we can have such a tool by combining diffusion MRI with biophysical modeling. So first I will try to explain how we can use diffusion MRI as an in vivo microscope, and then I will present the road to validation that we've traveled to really bring white matter models to a high level of confidence, and how we are now using these white matter models, for example, to study motor recovery from subcortical stroke. And I will wrap up with developing gray matter models, which is really the next challenge for the community. And it's also what's really, what my work is really focused on currently. So first of all, how is diffusion MRI an in vivo microscope? So if you take an MRI image, of course, the spatial resolution is very coarse, and you can have no pretense for microscopy here because the features we are interested in at the microscopic level are on the order of a few microns. Now, our luck here is that the diffusion process that takes place, so the water molecules that travel in this microstructure during the diffusion MRI experiment, they travel on the order of a few microns, on the same length scales as these microstructural features that we are trying to uncover. And their path here is highly dominated precisely by the microscopic features we are trying to retrieve, for example, membranes or other hindrances in the tissue. So the diffusion process is actually an excellent probe of tissue microstructure. Now the problem is that with diffusion MRI, we just measure this signal decay here as a function of increasing diffusion weighting. And the question is, how can we really relate this very featureless signal here to this very rich microscopy picture here? So the problem or the limitation of diffusion MRI is that it's a very indirect probe of tissue microstructure. And so this is where biophysical modeling comes in. So the biophysical model is a link between the MRI signal and the underlying microstructure. And the most important point in biophysical modeling is really drawing the right picture of your tissue, because any model is a simplification, and our goal is not really to render all the details or all the complexities of the tissue, but rather to tease out what are the most relevant features in this tissue and what can we say about them. So for example, in white matter here, we can assume that most of the water is either inside or outside axons, and axons can be modeled as infinitely long sticks. So inside axons, diffusion is unidirectional with a certain diffusion coefficient, and outside axons, you have two different diffusion coefficients parallel and perpendicularly to the axon. Then you have the compartment weight, so how much water is inside the axons versus in the extraaxonal space, and you can allow some orientation dispersion here for these axons along the main orientation of the fiber bundle. Now for gray matter, of course, we have to draw a completely different picture. So in gray matter, for sure, we have to account for cell bodies here, or soma, because they are quite present in gray matter, or in cortex rather, should I say. And then the neurite orientations this time can maybe rather be assumed to be isotropically distributed. And in gray matter, we don't have myelin. So myelin here really ensures water compartmentalization. So we can assume that there is no exchange between the compartments in white matter. But this may not be the case in gray matter. Maybe in gray matter, we have to account for compartment exchange here across the cell membrane. And so really the questions we have to ask ourselves constantly are, which microstructure features are truly relevant for the tissue at stake? Can I sensitize the diffusion MRI signal to those features? And most importantly, can I reliably estimate all the model parameters from the data? So this is the path that we've taken for white matter, and it's taken quite a few years. I will try to walk you through it fairly quickly, but it was, I mean, there's really a lot of work behind just a few slides. So in white matter, roughly, this is the standard model that I've previously described. And you can see you have a certain number of model parameters here. So you have your compartment fraction, your diffusivities, and here you can allow yourself some parameters for the orientation distribution function of these axons, depending to what level of complexity you want to parametrize it. Now, if you read the literature, there are really a lot of different implementations of the standard model, of course, each with its own specific acronym. And behind each of these implementations, the issue is that there are additional underlying assumptions to simplify the problem and essentially stabilize the fit. So popular assumptions, for example, are setting these two diffusivities inside and outside to be equal or to fix them to some value. Then this assumption here is the so-called tortuosity model that fully links the extracellular radial diffusivity here to the intraaxonal water fraction via this relationship. Or some models, for example, assume perfectly parallel axons because they cannot afford to parametrize the ODF of the axons in a more complex way. Now, the problem is that with all these assumptions, there's a potential loss of specificity for the remaining free parameters that you're trying to estimate. So for example, if in your disease there is some intraaxonal injury here that affects the intraaxonal diffusivity, well, if you fixed it a priori to some value, this change cannot be reflected in the model and will be instead absorbed by a different parameter which has nothing to do with the diffusivity. So you're really losing specificity, and this kind of defeats the purpose of doing biophysical modeling to start with. And so a few years ago, I started by really taking an honest look at this fitting landscape. And so starting from the full model here and giving myself 5 parameters, so the compartment fraction, the diffusivities, and 1 parameter to parametrize this orientation dispersion here. If you do a brute force fit, actually you see that in a very homogeneous white matter tract, for example, the distribution of values that you get from this fit is extremely broad, and very importantly, it's actually bimodal. So the conclusion is that this very simple model actually has 2 distinct mathematical solutions, both of which were actually biologically plausible. And roughly, they can be categorized as either the intraaxonal water is faster than the extraaxonal or the other way around. So this raised a very important question across the community. And it was very interesting that in the subsequent years, many different groups actually tried to answer then this question using very different methods or approaches. And so it was a very nice moment in the community that all these papers came out roughly at the same time. And so this is work from our group here at CIBM and a group in Denmark and another group in Germany. And so everyone used a different approach. But what was really striking is that we all came to the same conclusion or answer to this question, that the intraaxonal water was faster than the extraaxonal. So this is one additional brick of understanding that we brought to the white matter model. Now, the last issue we addressed was this tortuosity model, which is also often assumed to simplify the problem. And this had been shown actually quite a few years ago to break for tight packings, which is often the case in white matter. So actually, if you start from a very compact configuration here of axons, highly myelinated, if you have some axonal loss, so you're moving in this direction, actually your axonal fraction will be mainly affected, but this mobility here in the radial plane, not so much. Whereas if you have homogeneous or very widespread demyelination, you can see that very broadly your extracellular space is much freer now, and then this extraaxonal radial diffusivity will be affected. And so we've shown actually these two different dynamics of these two parameters in a mouse model of demyelination. So at different demyelination stages, how each parameter was affected, and they actually didn't correlate, so you could really obtain complementary information from each of them. And most importantly, we've also validated them against electron microscopy. So for example, here the axonal fraction derived from diffusion MRI correlated significantly with both myelinated and total axon fraction from electron microscopy, but not with the G ratio, so the myelination of the axons, whereas the extraaxonal radial diffusivity didn't correlate with the axon fraction, but correlated significantly with the G ratio. So the axonal fraction from diffusion is actually specific to axonal density, which is very good news. And then the extraaxonal radial diffusivity is specific to myelination. So now we feel that we're really trying to understand a little bit better our model, and we have more confidence in whatever we are estimating. So what I've been trying to explain is that in theory, biophysical tissue models can really provide specific metrics of microstructure, but in practice, the output is really dependent on the validity of the model assumption. So you really have to interpret the output with caution. And so all the validation work that we've put in showed that the axonal diffusivity is faster than the extraaxonal, so we cannot assume them to be equal or fix them to some value. And also this tortuosity model doesn't hold in white matter. And what's most concerning is that these two assumptions here are the assumptions underlying the naughty model, which is by very, very far the most popular white matter model that people use today. So I'm just suggesting that you may want to use this model with caution, or interpret the results with caution if you read a study that uses naughty or another model that you may not be familiar with, and with the assumptions. And so personally, I'm very involved in applications of this white matter model now to dementia, so trying to understand or characterize the white matter degeneration that undergoes in the dementia process. But actually, I won't be covering these applications today at all. And instead, I will present you preliminary or unpublished results from a very exciting collaborative project here we have at the CIBM on motor recovery from subcortical stroke. And this is work that I'm doing with Elvira Pirondini and Gabrielle Girard. And so this is part of a very broad project involving many actors and many methods. But essentially, this is on non-human primates, where a model of stroke is induced by causing a lesion here in the internal capsule, which then causes significant paralysis of the arm and hand in the monkeys. And then there's a longitudinal follow-up of their recovery using a whole battery of kinematic metrics, which I'm not the first person to explain. But roughly here, you can look at the number of reach movements of these monkeys per trial. So this is the pre-lesion level. Right after the lesion, of course, it drops, and then it partially recovers in following days. And then there are other metrics, for example, for the hand, the number of hand openings. You can see it actually plummets after the lesion and never recovers. So this is a very good way to characterize the lesion and its recovery from different parts of the corticospinal tract. And finally, the last part of the project that I'm actually involved with, as well as Elvira and Gabrielle, is the ex vivo assessment of residual brain damage from diffusion MRI. So here we start, for example, with macroscopic connectivity. So here, doing the tractography on the corticospinal tract. This is work by Gabrielle, of course. You can appreciate here on the healthy hemisphere, the very comprehensive and nice reconstruction of the whole corticospinal tract. And on the lesion hemisphere, here, so the lesion is identified here, you can see how crippled this is and how few streamlines now actually make it from the brainstem to the motor cortex. And this is also illustrated here on this coronal view, where clearly the lesion side is significantly affected by the surgery. And so then we can also wonder how the microstructure changes along this corticospinal tract. And so for this purpose, we actually have to use an atlas-based segmentation of the corticospinal tract, precisely because the lesion area was so crippled, we couldn't really use the tractography itself to delineate the corticospinal tract. So instead we're using atlas-based registration of where the CST should have been. And we're looking at how parametric values actually evolve along the CST from the cortex to the brainstem. So let's start with empirical metrics that many of you may be familiar with. So for example, here, fractional anisotropy from DTI, you can clearly see the lesion here, quite focal in the corticospinal tract. And if we look at the profile of FA values along the CST, of course you can see the FA is much reduced in the lesion area and a little bit downstream here towards the brainstem. Now, if we push the analysis to beyond the first order Gaussian approximation, we also estimate here the kurtosis tensor, so a measure of tissue heterogeneity. Looking at radial kurtosis, we can see actually the damage or the difference to the contralateral side is much more widespread, including towards the cortex. And if we look at the profile of this radial kurtosis, along the CST, we can see, of course, it's reduced in the lesion area, also a bit downstream towards the brainstem, but it's also reduced upstream towards the cortex. And this is not something that we could have picked up with DTI alone, for example. And now, if we move on to our white matter models, so actually trying to retrieve specific features of the microstructure that were altered by this lesion, we can see that the parameters that were mainly affected were the fiber alignment. So again, here, quite a focal lesion, and this alignment is much reduced in the lesion area and a little bit downstream, but not upstream. And this profile is reminiscent actually of the FA one, which makes sense because the FA is usually highly dominated by the presence of axons and how coherent or how aligned they are. And the other metric that was significantly altered in the lesioned hemisphere was the axonal density. And here again, reduced density in the lesion area, also downstream, but also upstream here towards the cortex. And so the decreased radial kurtosis that we saw on the previous slide is actually due to a decreased axonal density here in the corticospinal tract that actually, so this degeneration is actually not just focal anymore, but it propagated to much more remote areas than the lesion itself. And this is very useful information for understanding the process of injury and recovery in the subcortical stroke. So this is now the kind of level of detail that we can achieve without actually cutting off the brain, right? So this is all non-invasive. And of course, these brains here had to be done ex vivo to be done in our center, but these are measurements that are available in vivo as well. So now let's move on to the cortex, because it's nice to characterize the white matter, but of course you may also be interested in characterizing the microstructure of the gray matter for a number of diseases that also affect gray matter. And so the challenge here, of course, is that we have to start from scratch. So we have to draw a completely new picture. And one main difference is that in gray matter, there is really substantial intercompartment water exchange that we cannot neglect. And so it's very, the reason we cannot neglect it is that there is clear signature of this exchange in the MRI signal itself. So here, if you look at white matter, how it behaves at high B values. So if you increase your diffusion weighting, this is 1 over square root of B, so high B values are here. An impermeable stick will really follow a linear form here, as a linear functional form as a function of 1 over square root of B. Whereas, so this is of course the case for white matter, you can see it's linear, and this justifies the assumption of impermeable sticks for modeling axons in white matter. Now in cortex, of course, you see that you have this curvature, so you have a deviation from this model that can be largely attributed to exchange. And another signature of the presence of exchange is that if you apply a two-compartment model to cortex, and you do this at different diffusion times, so assuming, here, impermeable compartment, so you neglect exchange, you can see that the neurite fraction that you estimate decreases with increasing diffusion time. And this is also a signature of exchange, because the longer time you wait, the more water will have also traveled outside the axons, and then it doesn't really behave like diffusion in a unidirectional narrow stick anymore. So this is why we really have to account for exchange. And so luckily, there is already an analytical model derived for two exchanging compartments. This was derived by Kerger in 1985. And luckily, we can also probably introduce some simplifying assumptions of our own here, like isotropic distribution of neurite orientations, and thereby also isotropic extracellular space. So I actually have fewer parameters to estimate than the white matter model, but nonetheless, because of this exchange term here, the elementary response function is much more complex in the presence of exchange than it was in the case of impermeable compartments. And so here, you can see that these apparent fractions and diffusivities actually become functions of all the other model parameters. And you can really appreciate how unappealing these equations are. And you can also guess that this makes for a very, very unfavorable fitting landscape. So again, the fitting is challenging, and we cannot do it brute force. So we have to use some additional information or orthogonal measurement. And so my idea was to actually estimate the exchange time first separately, for example, from time-dependent kurtosis, and then inject it into the fit to try to estimate the other parameters more reliably. So the idea is that if you have Gaussian compartments, so you have no diffusion time dependence, your only source of kurtosis or of heterogeneity is the intercompartment heterogeneity, so the fact that you have two distinct compartments or more. And so with exchange, this kurtosis then goes to zero as time increases. And so you can use this functional form here to fit the exchange time if you measure kurtosis at several diffusion times, and then you can use it as a priori knowledge. And luckily for us, this assumption here, which is very important, is actually verified in rodent cortex at least. So here you can see that past 10 milliseconds, your diffusion time becomes, your diffusion coefficient becomes constant with time, and your kurtosis here decays with a form that can be very well fit by this analytical form here. And so I've done this actually, so we can estimate the exchange time using time-dependent kurtosis. This is actually a parametric map of exchange time in the rat brain. So you can see it's fairly homogeneous here across the cortex, but even in the subcortical areas here, such as the hippocampus. And roughly the exchange time I've estimated was 15 to 30 milliseconds, which is consistent with a few other studies that have attempted to estimate this exchange across the cell membrane as well. And then using this as an input into the Kerger model, so starting from our non-exchanging compartments, where the fraction was artificially decreasing with increasing diffusion time, we're actually now able to keep it to a constant level independently of the diffusion time. So this is already a very good indication that we are accounting for exchange properly. So to wrap up, I hope I've conveyed that biophysical modeling is challenging, but definitely worth it. For our field, validation is absolutely crucial. So we have to start from simulations, but also use histology. And more recently, also multiple diffusion encoding schemes, which can really provide very rich information. And so regarding the gray matter, or at least the cortex, we need to account for intercompartment exchange. So this is, of course, ongoing work. Also possibly for the presence of soma, which nonetheless makes up for at least 15% of the cortex volume. And then in the very distant future, well, maybe not, hopefully not very distant, but so we can hope to also look at more complex brain structures such as striatum or thalamus, which are really a mixture of white and gray. So there it's even more challenging, but this would allow us to really provide people with a comprehensive in vivo brain microscope. So that's it for me. I want to acknowledge mentors and colleagues at NYU, where I did my first postdoc, and then colleagues and students at EPFL, as well as collaborators, so Gabriel and Vera mainly. Thank you.
Thank you,
questions?
Thank you.
Okay, so thank you very much. I'd like to invite the attendees for any questions.
May I ask a question? So thank you. It was really a beautiful talk, really enjoyable. Just a question, maybe I missed, but what kind of echo time did you use and B factor? And is it possible to do similar modeling on clinical scanners?
Right. So for the first part, for the white matter model, it's definitely possible to do it on clinical scanners because, so for example, let's go back. The model I'm using here, even in the macaque, can actually uses the same information that you need for DKI. So for a clinical in vivo, you can stop, for example, at B2000 or 2500 with 2 shells, and that's enough to derive all the white matter model parameters that I've explained earlier. So this is definitely not a problem. For the gray matter model here, so again, for kurtosis, you have no choice. So you really have to stop beyond 2500 because that's really your radius of convergence of your Taylor series. So you should only acquire data up to B2500 here, but at different diffusion times indeed. So this may be a little bit more challenging on clinical scanners because they are not readily available. So you have to use either WIPs or modify the sequence to put in the diffusion time yourself, right? And then for the full character model, so here I've indeed exploited all the capabilities of our 14 Tesla system. So I'm going to B10,000. It's an open question.
But with what echo time, excuse me?
Sorry, the echo time here was roughly, so to go to a diffusion time of 40 was maybe roughly 50 or 55, something like this.
So it's still long?
Yes.
55. So what kind of...
14 Tesla especially.
Yeah. Okay, thank you.
Yeah.
I wanted to ask a question about the biophysical modeling and the 6 parameters. This was somewhere in the beginning. I understand that the solution with the 6 parameters was derived from some nonlinear least squares, right? But then you said that multiple solutions actually looked physiologically plausible. So do you know if someone tried to actually incorporate the prior knowledge into a Bayesian framework? Because it's generally difficult to use nonlinear least squares estimation when you have so many parameters, right? If you put the prior knowledge to the estimation of it, maybe the solution would be more reliable.
Yes, so there is a group in Freiburg who uses such a Bayesian framework. For us, in a sense, so once we have this a priori knowledge of roughly in which corner of the fitting landscape we should expect a solution, so like very concretely that we impose now that the intra-axonal diffusivity should be faster than the extra-axonal, then you can choose your fit initialization in that area. And this actually suffices to typically find then the same solution reproducibly. But you can definitely use a Bayesian framework, yes.
Do you know how correlated these parameters are?
To each other, you mean?
Yeah.
Yes, so this is also something that we've looked at. So the most difficult to separate are definitely the fraction and the intra-axonal diffusivity. So they have like the highest correlation level. The others are fairly straightforward.
And do you know whether in the estimation procedure here, the steps were in one dimension or in multiple dimensions? Because if parameters are correlated, you might be really trapped in like some local optima, right?
Yes, right. So we've actually looked at this landscape really comprehensively. I haven't shown it here, but we actually plotted the minimization landscape, and you indeed have, so even around each of these two distinct minima, you have an ensemble of solutions that are highly probable that follow some kind of really narrow and shallow pipe. So then noise can actually make you travel indeed pretty much anywhere along that pipe.
Okay, maybe we have another question because time is, anybody else?
Yep, Olivia here.
Yes.
Good morning, Ileana. Thank you for this presentation. I'm more a PET than MRI person, but I understood quite well your talk. It was very clear. I had a question about the NADI assumptions. So you said that the NODI used today was based on two assumptions that may not be as right as we thought. Is it the same thing in clinics? And is there somewhere in the world like people start to think about these two assumptions and maybe work on it?
Yes, so definitely in the clinic, it's the same implementation. So I think what really helped Naughty take off is also that they made their code available right away. And so everyone who uses Naughty essentially uses the same version of it. But yes, so there are now, well, there are a few groups or a few people who have started to raise questions, because mainly actually biologists and clinicians, because they found, well, results or trends using the Naughty model that kind of were counterintuitive with what they would have expected as clinicians or biologists. And so having a hard time actually interpreting these results from Naughty. But I'm not sure the awareness is as widespread as I would like it to be. So I feel like I'm trying to communicate this as best I can for the past 4 years. But I still, I mean, it's still very much used and not really questioned, simply because, yeah, I mean, it's a tool, you can readily use it, and if you're not an expert, well, it's very easy not to look at the details, actually.
Yeah, totally. Thank you very much.
Okay, thank you. There is one question on the chat, which is from Shavat Barga. Can you please elaborate about Karger model, if it's not too long?
Oh, right. So where was that? Here. Right, so Karger model, you're essentially assuming two exchanging compartments. And you can assume, I mean, this assumption here of isotropic distribution is something that we've added, but it's essentially the same picture that we had for the white matter. So you have one compartment, which is like a very narrow cylinder or a stick, so being either an axon or a neurite, and its surrounding extracellular medium. And so in this model, well, you have diffusivities for each compartment, but you also have an exchange term, so water molecules can cross from one to the other. And so it's essentially basically a very similar model to the previous one, only that now you're allowing for exchange. So this elementary response kernel here, so if these were non-exchanging compartments, this would be your axonal fraction with the intraaxonal diffusivity and your extraaxonal fraction with the extraaxonal diffusivity. Now, because of exchange, they each get a little prime here, and they become apparent. And so to account for exchange, you can see that each of them now incorporates all of the other parameters in the description. So this is, I mean, this was, again, derived a very long time ago. I could point you to the paper if you're really interested about this model.
Okay, great. So I think, thank you, Ileana, for the very stimulating talk.